We've saved the best for last. It's reproductive system time. The book goes through the male reproductive system first and then the female reproductive system second. So for the sake of simplicity, I'm going to follow the same order of things. Reproductive system, major function is reproduction. To get a sperm close enough to an egg that it can fertilize it. Let's talk about the parts. So male reproductive system, we're going to talk about the major organs, and then we'll talk about the functions of those organs. And we'll start with the testes because that's where sperm are generated. So testes are here. They are found outside of the main core of the male body and are found in this sac of skin called the scrotum. So the white part here is the testis, two is testes, and then there are tubes inside of here, more on that later, and those are continuous with tubes inside of this structure here, which is known as the epididymis. And then that epididymis tube is then going to be continuous with this tube here, known as the vas deferens or the ductus deferens. And you'll notice has a bit of a circuitous route here as it goes up and over the pubic bone on top of the bladder, behind the ureter. So this little thing right here would be is representing the ureter, and then around the back side of the bladder and comes around here. And this is where it joins with a gland known as the seminal vesicle. And then at this point, it changes its name. It's no longer the ductus deferens or the vas deferens. It's now the ejaculatory duct. From the ejaculatory duct, that is going to go, so there's an ejaculatory duct on both sides, right? Because there's a testis on both sides, and those join together in the prostate. So that's this, we've talked about the prostate in the urinary system. Your book, there's a food theme here. It says it's about the size of a peach pit that surrounds the urethra. So inside of the prostate gland is where those ejaculatory ducts meet and converge, and then eventually join the urethra. The base of the penis is another gland called the bulbourethral gland, and those there are two of those, one on each side of the penis. We'll talk about what they do soon. And then, and that duct also joins the urethra. And then the urethra is going to travel through the penis and out the penis. Those are our organs. As we will talk next lecture about the development of the male and female reproductive systems, you will see, but it's worthy talking about now because it's on my mind right now. So what is it about this crazy circuitous route that the ductus deferens has to go through? Well, ovaries and testes start as the same gonadal structures, and they either develop into ovaries in women or testes in men. And so they're both starting inside of the body cavity of a young fetus. And then those testes have to descend from inside of the body cavity because they're essentially the same place that ovaries are. And they do this descent out and over the pubic bone and down outside of the body. Why? Well, evolution has some interesting hypotheses about why all this happens, but it turns out that sperm develops better at a slightly lower temperature, body temperature, than our core body temperature. So there you have it. So different angle, and we're going to start talking about some structures that help regulate the temperature of the testes. So we have, here's the testicle. On top of it would be the epididymis, and then that is going to be continuous with the vas deferens, which is this tube right here. But in this spermatic cord that descends out of the body, you've got not just the vas deferens, but some other things. The testes are a living organ. They need a blood supply. So that's what you see here. The blue would be the vein, and then inside of that network of vein is an artery. And you can also see a yellow line in there. So there are nerves as well that go down to the testes and the scrotum. And then surrounding the vasculature system and the nerve and the vas deferens is a muscle known as the cremaster muscle, pointed through here. So this cremaster muscle here, and then that is surrounded by some connective tissue. And then outside of that connective tissue, you have some more superficial fascia. And then these pieces comprising the scrotum. And then within this skin here, there's, and this fascia, there is a muscle known as the dartos muscle. So what does all of that do? Let me remember what order my slides are in. So as we noted, the testicles have to descend down and out of the body. As they do that, they actually move through the abdominal wall muscles that make up the structure of your abdominal wall. So there are some rings, inguinal rings, and when you do the 3D lab through Grace University, that I put a link up there for, which I encourage you to do, it'll give you a much better sense of the three-dimensionality of both reproductive systems. What you'll see there are some connective tissue rings that don't line up quite together. One's kind of off to the side of the other. That's handy because if you have a hole in a muscle, things can slip through it. What you want to slip through, of course, is the testes and their blood and nerve supply. What you don't want to slip through are the intestines. So this is known as an inguinal hernia, where the intestines slip through those connective tissue rings, as you can see. It tends to happen more early on in a young boy's life as things are still growing and moving around. But as you can imagine, this usually requires some sort of surgery to fix, or at least some mechanical assistance. Okay. But men are prone to these inguinal hernias because all of this is actually going through the transverse muscle and the external obliques in order to have the testes outside of the body cavity. And they're outside the body cavity because spermatogenesis or spermogenesis, depending on which textbook you're reading, is the body making sperm. Genesis, birth, sperm. And that happens best at 34 degrees Celsius or 93 degrees Fahrenheit. And you are aware, right, body temperatures around 98.6 degrees Fahrenheit, although they're starting to rethink that most of us have a body temperature slightly lower than 98.6, but not quite 93. So to have optimal temperature for spermatogenesis, testes are found outside of the body. And there are some structures that help regulate the temperature of the testes. One of them is the blood supply. So you'll notice that this vein kind of makes this network that surrounds the inner artery. And blood is going to be flowing into the testes in that inner single artery, and then leaving the testes in this blue vein. So this is known as counter-current exchange. One fluid's going down while the other fluid's going up. Counter-current exchange. What are they exchanging? Heat. This blood, as it leaves the body, is going to be a warm 98.6 degrees Fahrenheit. Ideally, that blood in the artery, by the time it reaches the testes, has cooled down to 93 degrees Fahrenheit. How does that happen? Well, if we start down here, and this blood is indeed 93 degrees Fahrenheit. As it moves up through these blue veins that surround the artery, then the heat from the arterial blood that's warmer is going to transfer to this venous blood, and it's going to heat it up. So as the artery blood descends, it's going to cool. And as the venous blood returns to the body, it's going to start heating up. So countercurrent, and they're exchanging heat with one another. Pretty clever. This system is also seen in the feet of a lot of water birds. So the geese out on the pond, their feet never really get cold because they have this countercurrent exchange to help prevent their losing a lot of body heat to their feet that are swimming through the water. Dolphins also have it in their fins to help them not lose their body heat inside of their bodies. Countercurrent exchange. Oh, do you want one more example? I know you do. So camels also have it, but it's in their brains. So the camels were worried about getting their brains to be way too hot in the desert. And so they have a countercurrent exchange that as they breathe in air, it goes against the cavernous nasal cavity. That whole thing, because it's wet and it's moist, it cools the air, which is cooling the blood that's on the top of within their nasal cavity. And then that is directed to flow and have this structure around the artery that's feeding the brain. So it's helping to cool the blood that's going into the brain, which is very handy if you live in the desert. Fun. Cremaster muscle. So this muscle, when it contracts, it's going to shorten, which means the testes are going to rise closer to the body. When it's relaxed, the testes are going to be further away from the body. So depending on the temperature, the environment, if the testes need to be warmer, they'll be pulled up closer to the body. If they need to cool off, then that muscle is going to relax and the testes will move further away from the body. Same thing with this dartos muscle. When it's relaxed, the surface area of the skin is smooth and expanded. If, however, it contracts, then it's going to cause the scrotum skin to kind of crinkle and wrinkle up, and that's going to help maintain heat in the testes. So three structures that help regulate the temperature in the testes. A few years ago, teaching this lecture, and one of my students pointed out that this image from the book was wrong. And I said, in what way? And he said, well, I don't think testes are equally positioned in anybody. So it turns out there is a study done in 1960 by authors Cheng et al. And they surveyed 486 men and measured the position of the testes. And they found that for 27% of their sample, 20% of them had the left testes was higher than the right, 11% it was equally positioned, but for the majority of men, 62% of the men, the right testes is higher. So this diagram would only be correct for about 11% of the population. Why does it vary? Well, the thought is that the liver is on the right side, and somehow the development of that is pulling the right testes higher. But I think a lot of that's still unknown, and clearly it can vary, and it all just kind of depends on how things go during development. But as you can see, for the majority of men, the testes are not equally positioned. And as you might be able to imagine, it is handier to have them not equally positioned. And if you can't imagine why that would be advantageous, me explaining to you it, I don't think it's going to help you at all. So I'll leave that there. So let's get into the anatomy of a testicle. So the testes are actually full of tubes, and the names of those tubes are seminiferous tubules, and the job of those tubules is to make sperm. And As you can see, testes full of these tubes. If it's tubes, they're probably lined with epithelium, you got that. And then these are all going to converge into the epididymis, which is also, as you can see, a series of tubes. And then that epididymis is going to be continuous with the ductus deferens. Note, testicular cancer is the most common cancer for men between the ages of 15 and 35, which is why checkups are important for men regarding their testes, because there's a chance of having testicular cancer. My cousin had it. All right, more than you needed to know. Sorry about that. So some sperm trivia, because I get asked this a lot during these lectures, and this is what I put together for you. So spermatogenesis begins at puberty, which is around 13, 14 years of age. Before that, the testes are not making sperm. And a healthy male makes about 90 million sperm a day. They can make up to, I've read 400 million a day is an estimate for men that are in their late teens, early 20s, when you're in your high fertility years. Sperm are specialized epithelial cells. That's an interesting thing to think about, right? And it takes about 24 days for a sperm to develop in the testes. We'll look at that a little bit more, but at that point, even though it takes a little over 3 weeks for them to develop within the testes, they're still not mobile, so they can't swim on their own. So they actually have to be moved from out of the testes into the epididymis. They can't do it on their own. We'll talk about how that happens. So the sperm gain their maturity in the epididymis, where they gain their ability to swim, and that process takes about 20 days. So it takes a minimum of about 40, 44 days for a sperm to go from stem cell to sperm ready to get involved in the reproduction of humans. Scanning electron micrograph of a cross-sectional view of that seminiferous tubule, which is inside of the testes. So the stem cells, I just want you to get an idea of how this works. The stem cells for the sperm are hanging out here on the outer ring of the seminiferous tubules, and those are going to start to divide. And as they divide, they're going to start pushing the developing sperm closer to the center of the tube. So you can kind of see how these are changing shape as you get closer and closer to the center of the tube. And what you're seeing here are a bunch of sperm tails that then will be, remember peristalsis, right? In the digestive system that you have, it's kind of like milking a cow. If you've ever milked a cow, where you squeeze from one end of the tube and move the fluid through the tube, right? Same thing, the sperm have to be moved through peristalsis from the testes into the epididymis. There's another view. This micrograph is wildly colored. It's not actually that color, but also another interesting view of seeing these developing sperm as they come from their stem cells. So here it is in diagram form. On the outer edge of that tube, you have a stem cell called the spermatogonium, and those are going to start going through division, a process called meiosis. Meiosis is a type of cellular division that divides the genetic material in half. So the stem cell and most of the cells in your body, except for the sperm and the egg, all have 46 chromosomes. You got 23 from your mom, 23 from your dad, meaning you got 23 from the sperm and 23 from the egg. So when you're going to make more sperm or more eggs, then you have to go through that division again. You've got to divide the 46 chromosomes into half, so that when you take a sperm and an egg, you get 23 and 23 make 46, not 46 and 46 make 92. That doesn't make a very nice human. So you got to divide the genetic material in half, and that happens through a process called meiosis, where cells divide multiple times to reduce the genetic material by half. So while this cell has 46 chromosomes, this sperm has 23. Remember, at this point, can it swim? No, no swimming. Here's a progression of what sperm look like as they're maturing. They start as round cells, and as they mature, they start to grow this tail, also called a flagella. And as that flagella whips around, then the sperm is able to swim. There's this acrosome here, which is important. It has an enzyme in it that allows the sperm to helps the sperm to enter the egg. More on that later. And of course, the nucleus has the genetic material that's required in order to start new life. And lots of mitochondria, because as you can imagine, swimming takes a lot of energy, got to produce a lot of ATP, so you need a lot of mitochondria. So here's what a cross-section of a seminiferous tubule looks like underneath a microscope. And we've talked about this part, these seminiferous tubules, but there are also some additional cells inside of the testes. One of them are these interstitial cells. And what I want you to know about interstitial cells is they're not part of the seminiferous tubules. They're outside of them. They're kind of squished in between the tubules. And they're important because they make hormones like estrogen. Estrogen, a little bit of estrogen, but mostly testosterone. And testosterone is the hormone that tends to give, does give males features that we associate with maleness. And then there are also these myoid cells, and the myoid cells surround the tube, and you've seen that MY before, myosin, right? So those myoid cells act like little muscles that surround the cells, and those are the ones that are doing the peristalsis to move the sperm from the testes into the epididymis. So now we've arrived in this epididymis, which is a coiled tube. And if you were to take that tube and pull it out, you'd find that for each one of the epididymis, they're each about 20 feet long. And this is where sperm are stored until an ejaculatory event. Now, as we mentioned earlier, it takes about 20 days for the sperm to mature there. And while they're doing that, they're slowly being moved from the entry point to later in the epididymis, where these are in this portion of the epididymis, that's where we would have the sperm that are mature and ready for an ejaculatory event. Some student always asks me, well, how long can they live there? Having a hard time finding a clear answer on that one, but from what I've read, the best I've gotten is a couple of months. So maybe around 60 days or so, they can live in the epididymis. So this is what a cross-section of that epididymis tube looks like, a little different than the seminiferous tubules. They are lined on the inside with pseudostratified columnar epithelium. So works in other systems, slightly different function here. Instead of cilia, these have microvilli, similar to the microvilli in the small intestines. So lots of surface area. Well, what are they doing? Well, they're secreting food for the sperm. So sperm have lots of mitochondria, they swim, so they need some ATP. Those cells and there's really small cells, right? They're basically genetic material with some enzymes, some mitochondria, and a tail. There's not a lot of food storage inside of a sperm, so they require food, which is provided by the epididymis tubule epithelium. And in the event that there is not an ejaculatory event and the sperm die, then they need to be cleaned up for the next wave of sperm coming, because 90 million are made every day. So these cells are able to do phagocytosis. Well, what's that? Phagocytosis is basically, well, we talked about that with the macrophages that are inside of your lungs, where they have these extensions where they can reach out, grab debris, dead things, cells that need to be cleaned up, and pull them into their body. And inside of the body of the cell, there are these packed little vesicles called lysosomes, which are full of digestive materials. And that's how all kinds of cells in our body get rid of the waste products that are in our, or dead cells that are in our bodies. Handy. So the pathway of sperm during ejaculation. First thing to recognize is they're not starting in the testes, right? We've established that. They certainly get made in the testes, but they're not ready for ejaculation until they've spent about 20 days in the epididymis. And at that point, when they are mature, then they are ready for ejaculation. So ejaculation starts in the epididymis and then moves from the epididymis to the vas deferens. From the all the way around, and then from the vas deferens through the ejaculatory duct, from the ejaculatory duct into the urethra, through the urethra, and then eventually out the external urethral orifice. Quite a little journey those sperm go on. Anything else we wanted to talk about here? Nope. Cross-section of the vas deferens, or the ductus deferens. Same tube, different name. You can see the majority of that mass of that tube is made up of smooth muscle, and then it is lined on the inside with a stratified epithelium with a collapsed lumen, and sperm are moving through the vas deferens and the ejaculatory duct and the urethra through peristalsis. This peristaltic waves moves through that tube as the smooth muscle contracts, and the nervous system subdivision that's responsible for that is the sympathetic. So sympathetic responsible for contracting smooth muscle in a peristaltic wave for ejaculation. So let's talk about those urethral sphincters. You'll remember there's an external urethral sphincter and an external urethral sphincter. So give yourself a moment and recall, if you will, where they are and which one should be open and which one should be closed during ejaculation. Final Jeopardy. Okay, if you said that this was the spot for the internal urethral sphincter, you are correct. Because this is the bladder. Yeah, so bladder. So this little sphincter needs to be closed, right? We would rather that urine and sperm are not coming out of the urethra at the same time. So close that one. If you said the external urethral sphincter is down here, you are wrong. You didn't say that, you know, it's here. It's in the pelvic muscles of the skeletal muscle. This one needs to be open, yeah. All right. So what occurs during a vasectomy? Remembering that that's called the ductus deferens or the vas deferens. Well, the point of the vasectomy is to make sure that the sperm are not going on this long journey. So that procedure is usually done about right here. So an incision is made in the scrotum. Use your words, ductus deferens is found. It's clipped, cut, and then often soldered on either side so that that tube is no longer continuous. That surgery can be reversed and that they can get those two tubes to join and heal together again. Doesn't always work, but it certainly can. Important to note, does the testes continue to produce sperm after a vasectomy? Yes. Does the testes still produce testosterone after a vasectomy? Yes. The testes has no idea that this vasectomy has happened. As far as it knows, it still needs to produce sperm and just as much testosterone as it did before. The only difference now is that the sperm never leave the epididymis, and thank goodness for all that phagocytosis. So let's talk about the glands. We've talked about the pathway. There are some glands involved in the ejaculatory process, and they are those bulbal urethral glands. So here it is here, and food reference, your book says it's about the size of a pea, and you have two of those. There are two of those at the base of the penis. There's also this seminal vesicle or seminal gland, and the prostate that goes around the urethra. Function of those, well, let's talk about that. We'll start with the bulbal urethral gland because it does its jobs first, and its job is lubrication of the urethra. And when it produces its mucus, it lubricates the urethra. It's often called the pre-ejaculate, and it also helps to neutralize the acidic pH that's been left. If there are traces of urine inside of the urethra, that mucus has a very basic pH. It's going to neutralize that acid, which is handy because sperm prefer a basic or neutral environment rather than, at least neutral environment rather than acidic. The other two glands, the seminal glands and the prostate, contribute to the volume of the semen. So the sperm don't actually make up a whole lot of volume of the semen. Most of the semen volume comes from these two glands. Why does it need it? Well, sperm need food, as we talked about. So the semen is providing food for the sperm, and it also produces something called prostaglandins. And basically those act like hormones, and the sperm need aid in becoming mobile, in being the best swimmers that they can be. And those prostaglandins also help aid fertilization. So most of the semen volume comes from the seminal glands at the posterior side of the bladder, right? These guys, one on each side, back side of the bladder. And then about 30% of the volume is coming from the prostate. And they're both contributing different things that aid with the sperm to live long enough to do their jobs and to be activated, meaning they can swim. All right. Semen trivia. I know, you were wondering. So in healthy semen, there's 20 to 150 million sperm per milliliter of semen. If there's less than 20 million sperm per milliliter of semen, then that's considered a low sperm count. It is quite a journey that sperm take through the female reproductive tract, and few of them actually reach the egg. And because there is this protective coating around the egg that the sperm have to get through, you actually need more than one sperm to get there to help. It's a group effort to get one sperm into that egg. So less than 20 million, that's considered a low sperm count, and a couple might have fertility issues if there's a low sperm count. And this was also somebody's job. Somebody did a scientific study on this and found that on average, sperm leaves the urethral orifice traveling about 11 miles per hour. But the sperm are not swimming, right? That's the semen leaving, and that's through that peristaltic wave that is due to the smooth muscle contraction that is stimulated by the sympathetic nervous system. Sperm aren't doing swimming at that point. They do most of their swimming right at the end of their journey, actually. And how fast can a sperm swim? Well, about 5 millimeters per minute, or about 5 body lengths per second. So try to imagine that. And they live for about 48 to 72 hours within the female reproductive tract, which is 2 to 3 times longer than the egg can live. So an egg lives for about 24 hours once it has undergone ovulation. But sometimes we talk about, like, women, you have this week that you are fertile, 5 to 7 days. Well, but the egg's only fertile for 24 hours. Yes, but the sperm are, are alive for 48 to 72 hours. So if sex has occurred 2 to 3-ish days before the ovulation has occurred, then you've got a window of time wherein fertilization could occur. All right, but it's not all about ejaculation. We also need an erection. So the little blue pill, its technical name is sildenafil, sure, but it's commonly known as Viagra. So this is an interesting backstory if you're not familiar with it. It's that this was originally a drug that was trying to be investigated for blood pressure regulation. Because what it does is it helps to relax the smooth muscle in arteries. So you'll, I hope, recognize artery, capillary, vein, and then there are these little sphincters around this shunt between the artery and the vein. And when these are open, then the blood can flow easily through the capillary bed. And when these are closed, or that muscle is contracted, then the blood doesn't go through that capillary bed. And so, and these are found throughout the body. And it's one of the ways your body helps shunt blood in different directions. So, one of the issues with high blood pressure is that the blood vessels are too constricted, so blood flow is reduced. So they were trying to explore a drug that would help relax these muscles and the smooth muscles in the arteries and help with blood flow. So as they were doing the experimental trials for this, and they started asking men if they were having any side effects, some of them said, "You betcha I do." Let me tell you about what this is doing, and perhaps we could extend the study. No, I don't know if that's actually how it went. But so they found out that for regulating blood pressure, not so great, but whoa, it sure helps with erections. So let's market this little blue pill, and you know the rest of the story. So how does it work? Well, we need to talk more about the anatomy of a penis in order to understand that. So penis is not a muscle. It doesn't have any muscles in it other than the smooth muscle that's inside of the arteries. But the mass of a penis is connective tissue. So what you're looking at here now is this network of connective tissue, and then these blue portions here would actually be empty, right? So it's sometimes referred to as spongy tissue, and then if you look at underneath a microscope, it looks spongy. And all those little blue holes are what gets filled with blood during an erection. And there are 3 compartments to a penis, or 3 bodies. The 2 larger ones are known as the corpus cavernosum. So corpus meaning body, cavern meaning a cavern. And then, and these red dots here in the middle are the arteries that feed the blood to those corpus cavernosum. And then in the 3rd body is, which is the corpus sponginosum. This pink circle here is the urethra. All right, here's another view of it, just to get an idea of the collagen. It's everywhere. So what's making this network are these collagen fibers, and then the blood's going to flow in between these collagen fibers when the penis is erect and be filled with blood. So an erection is a vascular event where there's more blood going into the penis than is leaving. So how does that happen? Well, in order for this to, for the penis to have lots of blood in it, then these blood vessels are normally pretty constricted, and during arousal, those relax. And it's the parasympathetic nervous system that allows those to relax during arousal. So they relax, blood flows in, and as it flows in, as the volume of blood increases the size of the penis, then this connective tissue layer around the outside gets pressure placed on it, and you'll notice there are these 2 blue dots here. Those are veins, and those are the veins that drain the blood from the penis. So those actually get pressure put on them, and so traps the blood inside. Blood can't drain out until it drains out much slower than it's entering. And thus the erection is controlled by the parasympathetic nervous system, relaxing the smooth muscle inside of the arteries. But it also gets assistance from the skeletal muscle system. This red muscle that you see both in the male and female sketches here is called the ischiocavernosus muscle. And during arousal, it contracts and restricts blood from leaving erectile tissue. So in males, the erectile tissue is the penis. In females, the erectile tissue is the clitoris. And we'll talk more about the clitoris in our next lecture, but it is more than just this. There's more to it than that. But here's your hint. This ischiocavernosus muscle is here and here. And then in red here is another one called the bulbocavernosus muscle. And both of those, as they contract, and this one's the bulbocavernosus muscle, as both of those contract, I should have written them both up there, they are restricting blood from leaving the erectile tissue. And that allows for the erection of the penis as well as the clitoris. So that's our segue into the female reproductive system, which will be our next lecture. How do I end this one? Well, if any of that has been interesting to you at all, may I recommend you a fascinating read. So Mary Roach wrote Gulp, that had the story about Elvis that I shared during our digestive system lecture. She also has written a book called Bonk, The Curious Coupling of Science and Sex. And it's primarily about the science of orgasm in both men and women. It's a fascinating read. She does a great job of integrating science with humor in, I think, appropriate ways. Anyway, if you're interested, I recommend it. This down here, she also wrote a book called Stiff, which is about cadavers and how cadavers are used in a variety of ways in our society. Also a great read. She's a great author. Anyway, see you next time.